Hello, welcome to EASL Studio. This is your weekly hepatology news broadcast. In today's episode, we'll be asking a question: do we care enough about the importance of DILI? I'm Guru Aithal. I'm Professor of Hepatology, University of Nottingham. I'm pleased to have Professor Maribel Lucena from University of Malaga, Paul Watkins from Chapel Hill, North Carolina, Dr. Skip Hayashi from Federal Drug Administration, USA. Need to confess that we all have interest in DILI, so we can say that is our conflict of interest, so we think it's important. So we'll build on the discussion on how we identify signals better, how we diagnose it, how we assess causality, what the role of biomarkers are, what barriers for implementation of what we have already found and done and advanced so far. So let's start with why is drug-induced liver injury important? So I'll start with one of my speakers, Maribel. Why is it drug-induced liver injury important?
Well, thank you, Guru. Thank you for inviting me to be here sharing this session with all of you. I think DILI is important because DILI is unpredictable, idiosyncratic. It is very much host dependent. So reaction that occurs when a patient that is unique to the form that reacts to drugs is exposed to a drug or herbals or dietary supplements and develop an adverse reaction while the majority of people can tolerate it. So that makes DILI a very challenging process to be diagnosed. And I will say also that in recent years, since we have been very successful with the ability to develop drugs to eradicate viral hepatitis C, now there is hepatologists are being most focused on drug-induced liver injury. So I think that is very good. Also, new drugs, particularly used at the hospital setting, for example, immune checkpoints, biologics, and other compounds are being prescribed, and new forms of DILI are emerging. So that makes raise the interest of hepatologists and other scientists, other clinicians in drug-induced liver injury.
Okay, challenging that is why it's important, and probably the importance has gone up because something else is less in our challenging shortlist, right? Paul, why is it so important?
Well, there are, of course, a number of reasons. If you take paracetamol out of the equation, certainly irreversible liver failure from drugs is a very rare thing. It's not a major public health concern. However, abnormalities in liver chemistries, including all the way to jaundice from drugs, is far more common. And now it's true, when you stop the drug, the patient usually gets better, but there's always the question in the patient taking multiple drugs, are you sure which one is actually causing? So what it means is during that period of recovery, you may stop medicines that don't need to be stopped. And even worse, in some situations, when a patient develops liver toxicity, particularly jaundice, you're hesitant to try any new drugs either. So particularly in cancer, for instance, in oncology, when you get a patient who gets jaundice from a chemotherapy agent, that patient's cancer is going to go untreated. Now, they should recover when you stop maybe all their drugs. But I think the public health impact is often underestimated because people don't realize the toll of stopping medicines and not being able to treat the underlying diseases, including things like tuberculosis and developing multi-drug resistance.
Excellent. So it's the problem because you have given a problem due to treatment, but you can't even treat the underlying condition because you have to stop. And you have
in some instances, sure, yeah.
And I see Skip is just coming online. Skip, we were just saying that drug-induced liver injury is a challenge, and why is it important from the drug development side?
Yeah, well, since I've been here, I've been impressed with, first of all, how busy we are. Despite the fact that I think, I've always been taught that, the non-clinical data has gotten better and better at screening drugs out. Nevertheless, we are still seeing a fair amount of liver injury, sometimes not so bad, but sometimes pretty severe. And I can tell you we have had to put programs on hold. And that's obviously has large implications. And then, of course, we want to be accurate. We don't want to put programs on hold when they don't need to be, or not approved when they can go. So all I can tell you from our standpoint, we are only growing. And I think the fact that we formed a DILI team that's dedicated to that shows that. And I would like to grow it even more. So yes, we are very busy. It's very important to the FDA. Thanks.
Guru, can I make one comment
Sure.
out of that? Because I consult and have consulted for a long time widely with the pharmaceutical industry. Once you have a signal of potential liver injury, such as ALT elevations in a clinical trial, you can get into the dilemma of having to prove you don't have a problem, prove a negative. And Skip, obviously very fortunate to have somebody with his background to really judge these things. But we don't have any good way of telling a drug that, as Hy Zimmerman said, a dog that barks but doesn't bite. In other words, are the signals you're seeing really portend severe liver injury or just, a transaminitis? As a result, drug companies are hesitant to develop drugs once they see a liver safety signal, even though we know that many of them would be safe. So the point I'm making is drugs are abandoned at early preclinical or early clinical stages when liver safety issues arise because we don't have the tools to really tell transaminitis from a real important safety signal.
Yeah.
Okay. I mean, from clinical point of view, we are the ones who need to really identify it because many a times, like you are mentioning, that it's difficult to even identify it at the drug development stage, as well as identify the true signal at the drug development stage. But from the clinician point of view, the burden is irrespective of viral hepatitis getting cured. In UK, it's the second commonest cause of jaundice coming to secondary care hospital once you exclude biliary obstruction. So burden is still substantial to the secondary care. And its diagnosis is, as you are identifying, when you have a signal, you can't tell one from the other. But from the clinical point of view, actually just identifying a signal itself is not happening. I just recall one of the studies which was done in Europe about 10 years ago, where they looked at all the patients who were diagnosed as drug-induced liver injury for a 6 months period and identified the incidence in inpatients. And then took 1 week, they had an ethically approved protocol where they just identified every signal that reached the threshold of ALT elevation, which we call as a threshold for acute injury. And then a pharmacist and hepatologist went through all the signals for a week, went to the patient, and just by taking that signal and then going and finding out the facts, they could increase the incidence of drug-induced liver injury by 12 folds, which means that we don't even identify 90% of the signals. So this is the reason why I feel the recent publication from our friend Ayako Suzuki in one of the drug safety journals is quite important. She has developed an algorithm to identify drug-induced liver injury based on all the existing data. And this will probably be set up or can be set up as a signal where if you collect the data live and have the ask for the computer to identify the associated etiology, you can really identify drug-induced liver injury. So alarm system can be set up. So do you think this is feasible and should be the first step we need to implement or disseminate so that people can try out?
Well, I'll comment. Yeah, that study was a very important study. I think it's one of the first that I don't know, was successful. I think other attempts have been as successful as that one. And yeah, I do agree with you. It would be nice to have some sort of triggering for the clinician that when they go on to their EMR, they say, hey, according to this algorithm, you might be sitting on a DILI. Please check your meds, in particular this medication here. So, it's very sensitive. There'll be a lot of times when they'll say, no, it's something else. But yeah, I think that, and then you start looking at downstream outcomes. Did it shorten hospitalization? Did it create, cost savings and things like that? So yeah, I think you're right, Guru.
It comes up, yeah.
I think it
Maribel?
was a tremendous achievement by Ayako, because she is very committed and patient, because it took a lot of work to create that framework in order to identify those, because what she was identifying is drug-associated acute liver injury unexplained by other causes. So the important thing in the framework that she developed is that she was able to exclude non-drug causes based on the limited information of the electronic medical records, and also laboratory data that always is not as good at it. So it's relying on the information in the medical records. So that's something that we should also have to take into account. The scenario where the databases are being tested for this.
Yeah, the other message I got from that paper is pretest probability. I think 2 points in there is that if you looked at all the signals she had, it's about 6,000 or so. The highest pretest probability or prevalence of unexplained injury, which in inference is that this is probably due to drug, had the pretest probability of about 35%. It's the highest likelihood of all the etiology.
Okay.
And so we'll come to the other development, which is important, and Skip has contributed a lot to it, which is to develop an electronic causality assessment method. And we try and rely upon being absolutely exhaustive in our investigations to exclude alternative causes, irrespective of their pretest probability. In that study, just an example of pretest probability of new autoimmune hepatitis was about 1.5%. And drug-induced liver injury was 35%, but we have autoantibodies as an essential investigation in virtually every signal we see of acute injury. So at least we can have a pretest probability, which you would say, well, we didn't have before, at least in a one drug in that paper, might really give you a starting point as to say, well, what context we use what tests based on what? Because if you say ANA positivity, which is there on 20% of the population, and you have got pretest probability 1.5%, it won't make autoimmune hepatitis diagnosis more than 30% straight away. So, which will bring me to how do we use the revised electronic causality assessment method, and how would it improve our ability to diagnose drug-induced liver injuries? Skip.
Well, I wish you hadn't used the autoimmune example. I think hepatitis E would be a better example. The autoimmune thing gets into drug-induced autoimmune hepatitis, right? And that is something we need to keep an eye on, because I've seen it here and there is an intervention. I think that's the main thing. Unlike any other DILIs I really know of right now, there's some idea that you can intervene. So I think for that reason alone, the autoimmune markers might be still important. But hepatitis E is a big issue, because I know, I've been taught by all of you see more Hep E than we do. I mean, it's vanishingly small in the United States. So this is a big problem. I've gotten a lot of complaints from the, on our side of the Atlantic that Hep E is, that pretest probability is virtually zero. And so that needs to be worked out. And there are other things. There's a paper in Hep Communications that from the Asians, the Chinese, and they said that our stringency for Hep B core is off for them because all their patients, have Hep B core hanging around, and we sort of take some points away from it. So that also, and I'm thinking more along the lines of geography and where you are sometimes in pretest probability. But I think where the DILI is starting to work on having to modify the RECAM for certain situations, and I've thought about incorporating in these ideas of pretest probability, like giving people the option to say, if you're not in a high prevalent Hep E area, then we're not going to penalize you for that. So those kinds of things. So it's extremely important, though. Yeah.
Yeah, I think coming back to Hep E, I mentioned this second cause of hepatocellular jaundice in UK. In that study, if you put all the viral hepatitis together, you still wouldn't reach 10% prevalence in people presenting with jaundice. So of that, I think hepatitis E was 2%, while the others were less than 1%.
Yeah. But so Hep E is still your leading cause of viral hepatitis around
of the viral ones, yes.
Yeah. So we should, like you said at the beginning, maybe we should drop Hep A, right? Because it's so unusually.
That will depend on the geographical area, because in the adjudication committee meeting that we have many, reasonably a number of hepatitis E that as a cause of exclusion. So it's not that rare in Spain. And I thought in UK it was rising the frequency also.
None of them are rising. They, like I mentioned, acute viral hepatitis is simply very rare cause of jaundice presenting to secondary care. So context of use is, most of the time, like we mentioned before, that drug-induced liver injury, most of the research, most of the findings come from clinical care, clinical services, because they're uncommon in drug development. So we need to take our context of use as much into consideration when we are developing tools, diagnostic tools, as well as the qualification of them. Coming back to the point, Paul, where do you think clinicians could and should use RECAM for their diagnosis and improving diagnosis? Because despite interest and knowledge, we do know that we don't really have a specific biomarker for drug-induced liver injury so far. So we'll come to that in a later stage. So do you think there is an application there, or how do you disseminate your tool?
Yeah, no, it's
I'm sorry. Which Paul are you talking
sorry,
about?
No, so Skip. Skip,
wait, Guru.
Continue. It's you're the RECAM. Go for it. I thought it was for me.
No, so it's so first of all, when you talk about context of use and how we developed it, we also have to keep in mind that this was based on mainly tertiary and secondary care centers, right? So you have to think also about the primary care doc who may want to use this tool, and they may see more viral hepatitis than we know about, right? They will make that diagnosis. And so I think that has to be considered too. And so that's what the challenge of any of these tools, RECAM and RECAM, is it for the patient that finally gets to a tertiary care center, or is it for the primary care doc who's out in rural somewhere that's seeing the patient, for the first time by, they're the first contact. So, but how did you, to answer your question, I think it's not unlike the RECAM in some ways for the common clinician, is that it helps them look at the tests they should get, how to do the workup, and if they so want to generate the score, that's fine. But I think one thing I worry about is when I see a non-hepatologist, non-gastroenterologist trying to work up abnormal liver tests, they often don't do certain tests that are very, very important. So that's the way I
it's a checklist, which is a good one to really have. Yeah.
Yes.
Paul, you were making the same point, or something different?
Well, yeah. I mean, I think that, the RECAM and the RECAM are very helpful. And I know Neil Kaplowitz says he used to carry around the RECAM in his pocket to make sure it's a checklist. One of the things that unfortunately is very difficult and has not been successful to get into those instruments is the observation that drugs that cause DILI tend to have a clinical pattern, a signature, augmentin, more cholestatic in the elderly, more hepatocellular in the younger. And that is really what is the difference between expert opinion and these diagnostic tools. And the best source for that is the LiverTox website. If a clinician is faced with somebody who's got abnormal liver chemistries and he doesn't know which drug it is, the RECAM is the best right now to have a systematic checklist, but also go to LiverTox and look what those drugs typically look like. Now, these signatures that drugs have are good guidelines, but as Willis Madry famously said, drug DILI has signatures, but sometimes the signature is illegible. I always love that thing. And the idea is that just because you don't see that typical augmentin, mixed cholestatic injury 2 weeks after the drug was stopped, and the elderly, doesn't mean it's not the drug, but it's extremely useful, I think. And it's the difference between just using an instrument and what we call expert opinion, because that aspect we've not yet been able to figure out how to put into some sort of tool.
I think that the point by Paul is very important because he's raising also one important issue with that. It is the importance of careful monitoring of the patient and a meticulous recording of the data completeness before applying for any causality assessment method is absolutely important. But I think that even for physicians in primary care, they should provide DILI causality scoring, because that gives us an information on how confident you can be on how the data that they have handled is allowed us to attribute with certainty or with confidence the causality between the drug, the strength of the association between the drug and the appearance of that liver injury. That is important because it's not the same that they say highly likely, which means that maybe the patient have a re-challenge, or it's possible because other potential causes are also probable or possible, or maybe there is no information on the challenge. And I think so it's so important because I agree with you. What we do always is check the information, check the data, the completeness of the cause of the reaction and all the information, and then we decide this is drug-related or not drug-related. And then it's so we first apply expert opinion, and then later on a causality assessment tool. Now we are applying RECAM because, as I said to you, it's very important to give a scoring. Because also
confidence in
not to
diagnosis.
forget.
That's what you're saying.
Yes.
Confidence in diagnosis determines the action. Okay. We will come to preventing drug-induced liver injury. I think there is a potential for us to prevent a number of drug adverse reactions. This is been really a crux of pharmacogenomics, and we have, I would say, in UK made substantial progress in discovery and implementation. So I would just like to explore how we might use as it is. Is there any role for drug tests, drug genetic-based tests in diagnosis, or you think it's only for prevention? So, Paul.
Well, you're making, well, let me back up. So I think all of us went into collecting cases, getting DNA and doing genetic analyses with the hope that there would be a common set of susceptibility factors that you could put on a diagnostic chip commercially available. That's turned out not to be the case, number one. Number two, the actual predictive value of the tests we have now are not in the range we would like to see, or that would make a label, at least currently. the strongest associations have been with HLA alleles, risk alleles, which are the molecules that actually hold up the antigen, either class 2 from the immune cells or class 1, presumably from the liver cells themselves. And unfortunately, they've not been strong enough yet to for any drug that I'm aware of, or at least that's with lumiracoxib, which I know you know well, Guru. There was an attempt to get the drug back on the market with HLA typing, but so far that's not, as far as I know, been used to actually protect patients, to identify susceptible individuals. In theory, it could be done. And as you know, with Augmentin in collaboration with the European investigators, including Maribel and Guru here, we have found 5 independent risk factors for amoxiclav liver injury, right? And when you put them together, you can get a score. And if somebody has all 5 of these risk factors, they have a 1 in 100 chance of getting DILI if they take a course of amoxiclav. If they have none of them, it's less than 1 in 5,000. That might be useful if you were talking about an anti-cancer drug or something where there were a lot of alternative drugs you could use. But again, that kind of genotyping is not available commercially right now to do that. But it is showing a direction where using multiple genetic factors might have a role. You know, what I'm seeing it now is, you know, when in a clinical trial, if the patient develops jaundice and they took Augmentin, well, drug companies may want to go to the trouble of getting all 5 of those alleles to bolster the case it wasn't their drug. In fact, it was more likely to be Augmentin. But it's not moved into the clinical area, and I don't think we're actually close to use it for risk, you know, prevention right now. But Guru, I know you have a lot of experience in this area, and Maribel, take it away.
Yeah, I mean, from my side, one point which I want to make about diagnosis, sensitivity and specificity as HLA 5701 is much as good as IgG for autoimmune hepatitis. And constantly people talk about cost effectiveness and cost of autoantibody profile. We do about 40 a day minimum in one place is 20 pounds, 24 pounds, while cost of doing HLA typing specific is about 30 pounds. So we're doing 15,000 autoantibodies, which have got lower predictive value and many a times almost costs as much or more. The cost effectiveness studies have been done for prevention. Prevention-wise, UK has really made progress in terms of policies. Started almost about 15 years ago with the government highlighting that genetics is important and need to bring it into clinical practice in a broad level. And 2011, Chief Medical Officer setting out her annual report, calling it a generation genome and making pharmacogenomics as a part of that drive. And then Department of Health funding 100K project, which was aiming to sequence 100,000 people, but ended up in doing it in more number of people. And now we are in a phase of implementation where genomic services are implemented. Almost 7 GMSAs, Genomic Medical Service Alliances, have been set up. And they have got 3 or 4 big agendas. First one is about rare diseases, make what is called as patient odyssey shorter, so that they don't have to go from place to place to get a diagnosis. Second is cancer, pharmacogenomics in cancer, especially improving efficacy, identifying people who are responsive to particular treatment. And then third one is pharmacogenomics for pre-prescription, if at all possible. For that, PREPARE study was very, very interesting. You saw the results of that, which had 12 genes, 50 variants. They reduced adverse drug reaction in about 7,000 population by about 30%. And And in the follow-up phase, which wasn't part of the primary endpoint, there was further about 13% or so adverse drug reaction prevented. There are some retrospective cost effectiveness analyzed in US actually. If you implement it, I think the case control study may be about 30,000 people in there. You would reduce cost by about $5,000 or $6,000 per patient, something along the line. So evidence is there, but I feel that there is a threshold of action seem to be set higher for pharmacogenomic tests compared to other areas, including drug itself. You know, how many drugs have got cost effectiveness data before they're licensed? I'm just asking a question.
Well, I don't think there's cost effective. Yeah, I don't think we have cost effectiveness data before they go to approval. I doubt that, of course.
Exactly. So I think tests in themselves or diagnostic themselves are asked of more evidence than many other areas, which comes in a way of implementation, which might actually in the long term be cost effective for the whole society or patients or health services.
Guru, the one thing I would say is that I'm not exactly sure what your threshold was for these reactions. It kind of gets back to what Paul Watkins was saying. If it's a reaction that's not going to be that bad and the liver will adapt, then have you really done a service? You may have prevented that reaction, got cost savings, but did you take a drug away or did you do something that patient could have benefited from, right?
Yeah. So the study design, I'm just coming back to the study design. I won't go back to the retrospective cost effectiveness analysis separately. Just in that study, you had to prescribe and then within 7 days, randomized, the individuals randomized will have the results delivered, and then you change the dose. There are what are called the actions, which were the results were coded, and patients carried a card, QR code, and that will be straight away take you to the physician to a website where list of drugs and actions they could take was offered. And it was physician's decision to really change the action, which might be changing the drug at the time of when they knew it, which means that patient would be exposed to less than a week. They did deliver the whole panel in the week period. Second, they might really change the dose. Third, they switched the drug completely. So all the actions were their option. And then how did they measure the adverse reaction? This was actually patient-related outcome measures. Patient reported how did they feel at the end of the trial. And interviewers were not really part of the clinical investigation team. So this is as much an implementation study as you can get.
Yeah. But with regards to DILI, HLA alleles testing, which we know has been the most important contribution of this international collaboration to the knowledge of the risk of DILI development, I think it can be very, very important in some situations to reinforce a diagnosis, particularly when two drugs are given. I recall one case with terbinafine, and the possibility of an autoimmune hepatitis was being considered. Biopsy was inconclusive, and we test for the allele of risk, and the allele of risk was positive. And in the general population, that allele of risk is less than 1%. So we favor the diagnosis for DILI due to terbinafine. So that is a very important contribution. But my point is that, as Paul has mentioned, there are many initiatives in Europe, induced in the US, all of us in Europe, and encompassing many countries, the Global Alliance for Genomics and Health, or the 1+ million genomes, so that That means that a lot of information, the genome sequence data, is going to be included in the health record of a patient, and the physician in charge is going to be able to get all that information, that big amount of information, at the click of a button. So the important thing now will be how we translate the relevance of those HLA data, for example, to prevent the selection of a potential drug in a patient who has an allele of risk, not only for DILI, but also for severe adverse drug reactions. So that is a very nice opportunity to inform clinicians, to make them acquaintance with all this knowledge that has been gathered and generated thanks to this international collaboration.
Paul, is there anything such as this in US?
Yeah, there's a All of Us initiative the NIH has started, which is going to be 1 million patients, if they do what they say they're going to do, but they've already started, complete access to electronic health records and whole genome sequencing. And actually, there is an investigator, Russ Wilkie, actually in the US, who's already started mining what they have for potential DILI cases and looking for associations. So that exists. The one thing I did want to just make a mention of, since you brought it up, Guru, which is that, you know, these genetic risk alleles, you know, aren't perfect, but they actually do stack up very well to what we use anyway. So in the gastroenterology paper where we propose this 5 allele genetic risk factor, we also look to see what does that add to the clinical diagnosis. So we know, for instance, amoxiclav tends to be in an elderly man getting a prolonged course. We know about what the latency is. We know it's cholestatic mix, and we use that to make the diagnosis. So when we looked at latency, male sex, the duration of drug dose, et cetera, in a multiple regression analysis, we found that the genetic risk score was an independent risk. It was an independent predictor, and it was a stronger predictor than any of those other clinical parameters. So the genetics was actually the most single predictive thing to make the diagnosis when we compared the amoxiclav cases versus all the others that are in the DILI network. So I think we're moving towards acceptance, or at least being able to show utility in diagnosis.
Thank you. Thank you very much. I think we are getting close to our time. So I'll summarize. We have had an interesting discussion. It looks like common feature is really the science and technology, and especially a digital technology. It looks like we have got an algorithm, a computerized algorithm. We have got a electronic causality assessment method, and then hopefully we will have a digital records with our important pharmacogenomic data. Thank you very much for the interesting discussion. I will announce the next week's episode will be a long-awaited breakthrough, which is centered around the first drug which is approved for MASH. So you will have a next episode. Otherwise, it is up to me to now thank Paul, Skip, Maribel for your time. Hope you will be willing to answer questions if something which came along in the chat. Thank you.
Thank you.
Thanks you. Thanks.
Thanks, everybody.